think we need a new chair.
Hey everybody, welcome to Q&A with A&V. I'm Vincent Racaniello, and joining me from New York, Amy Rosenfeld.
Hello, Vincent. How are we?
I only saw you an hour ago, right?
Yep. We have to start off with our thank you to Emily and Kyle for the lovely bottle of wine that they sent. I think that was her name. I can't wait to drink it.
Yes, very good. Amy deserves multiple gifts.
I don't deserve multiple gifts, but it was very, very nice of them. And just because I've had a sore throat and stuff for the last, 5 days?
But you sound good now. Your voice is normal. Did you take something like tea or something?
I have sour balls.
Okay. Before we start, I would like to show 2 pictures.
Is this the incubator?
Yeah, it's the incubator.
Excellent.
The incubator is progressing, so this is the construction is done. It's got its paint and the rug, as you can see. So this is the
Conference room.
It's the conference area, right? So there's a little kitchen here. There's some space here, maybe, I don't know, for an office. Maybe we'll put Dixon in the back. I don't know. This is the office for our administrator. Then in the front, there's a big space for conference table. This door is very important here. This is the electrical closet. Now that's where the internet comes in, and that's what I've been working on this week. I can't tell you how much internet is in New York City. It's crazy. Here in New Jersey, I get 1,000 up and 1,000 down for $170 a month. And what I'm getting here, well, I'm not even going to tell you. So that's one part. And then the other part, this is the studio part. I kind of like the paint and the rug and the LED lights. We swapped all those out. So this is the studio. Now this, I'm going to cover all this. This door remains locked. We're going to take the handles off. Nobody comes in here. The other door will be the main entrance. And I don't know, I'll put some equipment there or something. I might record on this wall. I'm not sure. I might record on the other wall. But now, now that the internet is in, we need to get some furniture, right, Amy?
Yeah, we're going on Saturday.
So we're going to go Saturday. I need a couple of tables to record from and to work at. We need a conference table. We need chairs. And I want to get a better lock on the door. I'm spending this week also dealing with that. Amy sent me her locksmith. Yeah. Amy sent me her locksmith. His name is Popeye.
Yes.
And
that's good.
He was very helpful. He can't do it, but he's going to advise me. So that's the story of the incubator. Thank you for your support. That will be where we make even more science content in the future as this pandemic of the unvaccinated continues.
Yep.
Right.
Yep.
All right.
Apparently we need to continue.
So Steph has red fever, apparently. And she said great. she was surprised at the pipetting. And I said, in the old days, everyone mouth pipetted. And this is what she wrote.
I know how to mouth pipette.
Shocking. Do you think she learned that from you?
I think so. But I used to mouth pipette, and I'm 20 years younger than you at least. Yeah.
Right. The other thing that is dominating our conversation here is Tony Fauci and Rand Paul.
And, the geniuses are back at work. So does this count as a sporting event? So can I call the sporting agency and make bets?
Yeah, of course. You bet you can.
Okay.
I was called by a Newsweek reporter today wanting to know who was right, Tony Fauci or Rand Paul. So
well, definitely not the guy from Kentucky.
So she sent me a paper. She sent me a paper which came out of Wuhan and EcoHealth Alliance and Lin Fa Wang in Singapore. And it basically shows you take a bat virus. It's called WIV1 or 16. They're two different bat viruses that they had in the lab for a while. These bat viruses already infect human cells via ACE2. If you replace the spike gene with that of a variety of genes acquired from bats in a cave outside of Wuhan, they wanted to know if these spikes could mediate entry into human cells. Those WIV1 and 16 still enter human cells. So they have not been given any new properties. So it is not gain of function. I don't know what Tony's reasoning was. I don't know why Rand Paul thought it was gain of function, but it's not. And it's because they didn't change any fundamental properties of those bad viruses.
Well, Rand Paul is not a genius.
What did you say? He's an ophthalmologist? So he went to medical school, right?
Yeah, he went to Duke Medical School, and he's an ophthalmologist, and his wife is famous for having chickenpox parties even today. Fauci misdefined gain of function as a phenotype that is acquired through passaging. So that's a little off. But it's appalling that somebody who went to medical school and took genetics does not know what a gain of function is. Because we learn about it in genetics because there's many human diseases that are gain of function acquired. There are mutations that were gain of functions.
You know what else is interesting, Amy? When we made transgenic mice for the poliovirus receptor, that was a gain of function for the mouse.
Yes, I know.
People don't think of it in terms of mammals, but it can be for any organism on the planet.
Yeah. Well, that's what I just finished saying. It's for any organism on the planet because there are many genetic diseases. I think somebody in my family's son has too many folds in his brain, so it's too gyrated. I believe that's mediated by a gain of function.
Oh, interesting.
A function mutation.
Okay.
Whereas having lack of function, you get smooth brain.
Anyway, this is the link to the paper that has been discussed where Rand Paul said it was gain of function. It's not.
Maybe he should read the paper.
I'm sure his aide said, hey, this paper is gain of function. And so Rand went right up to the desk and said, this is gain of function. I mean, I wouldn't do that.
Well, one might want to fire that aide then.
I mean, when the Newsweek lady sent it to me, I read it. I read it with her on the phone, actually.
He's busy being a politician.
Yeah, I understand. All right, so that's the paper. Let's see what else we have here. I just, there was a recent paper on myocarditis in both vaccinated and COVID. Wonder if you saw it. Did you see it, Amy?
It does not sound familiar, but I can go through my folder later.
Amy will find it if she hasn't, because Amy is the great literature scanner. It's amazing what she can find.
Well, it's not that I just scan the literature. I actually read the papers.
I didn't mean to impugn that you just scan them. Of course you read them. By the way, double treat for everyone this week. The midweek TWIV is me and Amy.
And we're very tight. It's an hour, like 3 papers, very tight discussion about the 3 papers.
Yeah, 3 good papers. Everyone else bailed out, so Amy was on anyway to do it with me. Excuse me, but we had a tight discussion because Amy said, I have to get back to work. So, okay. Thank you, Amy, again. It'll be out at midnight. So after this live stream, I have to go edit it.
It was good. At least 2 of the papers were really timely for us, right?
Yeah, they're excellent.
Yeah. The 3rd paper was just because we're run by the public health and they need to go back to school.
All right. Are viruses like polio still circulating in the U.S. but not causing disease? And so you think they're widespread? What do you mean by viruses like polio? Does Manish mean enteroviruses?
Yeah, I don't know what that means. So if he's talking about that family of viruses, they're the most common human pathogen. Enteros are the most common human pathogen. And which disease is what disease is he talking about? Because polio causes like a febrile disease, and very infrequently it causes poliomyelitis. So what's the disease that he's talking about?
Yeah, there are a variety of diseases caused by enteroviruses. But I think since the U.S. switched to IPV in 2000, we probably have poliovirus strains circulating, right? Type 1
Even
most likely, right?
before,
Yeah,
even
probably even
before
before.
the
But
switch.
we don't check stool of people for polio, but if we did, we'd find it.
Well, you don't need to check stool at people. You just need to check the sewage water.
Yeah, that's true. You could do that. And, our friend over at the new school could do that. She has sewage.
Yeah, she could do it,
What's her
but she's
name? Can
busy.
you remind
Davida?
me?
Davida Smith?
Davida Smith. Yeah, she did this SARS-CoV-2 in seawater, sewage water.
Yeah,
Wow,
but
that
I
would be
think she's
fun.
moving to Texas A&M.
Well, we could still get this. The guy at Queens College has the samples. We could get them, but you have enough to do.
For the
Anyway,
moment.
by the way, so polio is circulating within the U.S. and many enteroviruses. Just talk to Amy, and she'll tell you all of them that are circulating and
There's
cause a
more than
variety.
110 non-polio enteroviruses that we're concerned about, plus the 160 rhinoviruses, which now are enteros. So what is that, 270? So there's 270
Yeah,
non-polio enteroviruses
and
that I'm
and
interested in.
and Amy just wrote and submitted a grant on this to try and continue the lab's funding. A
Yeah,
very
I'm
extensive
excited.
grant.
I'm very
Okay,
excited.
it's very exciting.
And it was a good project. I mean,
It's a
it was
great
good data.
project. I mean, the project is great. It's a great idea. We're generating data. I'm very much looking forward to doing it.
Yeah, we'll
Okay,
see.
H. Zoo. Could you talk about how exactly gain of function is defined? Any TWIV? I don't think we did a TWIV. Do you remember, Amy?
We did the NSAB with Michael Imperiali and members from that panel.
That was in the context of the avian influenza H5N1 gain of function experiments, right?
Yeah.
Yeah, so you could do that. Michael Imperiali, you could search for that TWIV. So here's the thing. I am making a video, but as Amy will say, good luck with that.
That's the wrong phrase I use, but that's okay.
Because until I get the studio set up. But short thing is gain of function is when you give any organism a new property of some kind. And like I said, our transgenic mice, we gave them the polio receptor. It's a new property. It's gain of function with mice. You can do it with yeast, fungi. You can do it with bacteria,
It's
viruses.
natural.
It
It's
happens
natural.
naturally.
It occurs in genetic diseases. They're natural. Not all of them are loss of functions. Think
Yeah.
about interferonopathies. That's too much interferon, right?
Yeah. So yes, they
Something
occur.
went awry.
They occur naturally. They occur in laboratories. If you stopped all gain of function research, a good fraction of research would stop.
95% of it.
So gain of function is not just something perceived as bad. So it's much broader than that.
It's just
so
unfortunate.
it's very unfortunate. But as usual, science is being misunderstood, right?
But the thing is it really came to prominence with Fouchier and Kaoka, right?
Yes, absolutely.
And it was pushed forward by somebody who's also, I believe, an MD, right? Isn't Relman an MD?
Yeah, David Relman, yeah.
So again,
also,
did they not go to genetics class? They were all sick on the day that gain of function mutations were discussed.
they had an agenda, man. They had an agenda. You know that, right?
I know. Because my genetics class as undergrad, we discussed gain of function. But I had two very good professors for undergraduate genetics.
So, the whole idea of someone in Congress arguing gain of function is absurd. All right? It's not arguable. There's a definition, and that's it. Although, according to Amy, Fauci has a slightly different view, right?
Well, his is that you have to passage something to make, and it's acquired
All
that
right.
way.
Yeah. So
And that's not really correct.
no.
Um, it's anything that has a different property than what the wild type that you're comparing it to has, as I
So
just finished saying.
So, Amy, if two coronaviruses recombine in nature somewhere, and a new property is a feature of the recombinant, is that gain of function?
Yeah.
If I want to make a different tasting beer and I modify the yeast to do that, is that gain of function?
If you give it a new function, if you make it so that like it no it you know, let's say it didn't metabolize lactic acid and now it does, it's gain of function.
Yeah. So there you go. Okay, here's a good one. Why are so many people getting the number wrong by mixing up infection and disease or mild disease? Seems like the studies of waning vaccine immunity over time lines up with your prediction. Why are people confusing this, Amy? I think you have a good handle on this.
Really? I have a good handle on this, really?
Yeah,
Uh,
I think you do.
uh,
They don't know science, right?
I think that they don't know what the words mean.
The thing,
And I think that there's a lot of misunderstanding, and they don't know what the words mean, and they don't actually know what vaccines do and what and stuff. And I think it's partly a fault of the colloquial definitions of the words are not the scientific definitions of the words. And people use them, and therefore they're used improperly. That's pretty much.
I think some scientists misuse them, and when they talk to the press, then the press doesn't know, so they just repeat it, and then
Well,
there it
yeah, a
is.
lot of scientists misuse them. But a lot of scientists also are speaking, and this is not their area of expertise. Although there are many whose this is their area of expertise, and one does have to wonder why they haven't returned their PhD. But that's a separate issue.
Where do you put it? Where do you return it when you have to return your PhD, Amy?
You send it back to the university and you say, unfortunately, I need to return this. What do you think? It's like, what do you think? You return it to Saks? No, you
Saks Fifth
send it back.
Avenue. Is that your
Yeah.
favorite store? Is that your favorite store?
No, Bergdorf says.
Barry wants to know if GSK monoclonal is available.
I believe it is.
So,
I believe it
Amy,
is available.
It's made by GSK and Vir Biotechnology. Is that
Yeah,
Virgin?
that's Virgin.
Is
Yeah,
Vir,
it's the
is Vir's name? Vir is Virgin?
No, it's virus biology. It's virus
I was gonna say,
biotechnology.
if he named it after
No.
himself.
No, didn't found the company. He just was hired as their, or maybe he did found the company, but he is really just now their chief scientific officer since he left WashU. But yeah, it's with him and Diamond and Wayland and this company, and then yeah,
Got it.
and it's actually supposed to be the best one.
Yeah, the monoclonal is very good. And
Well,
it
it also lasts a year.
lasts a year.
It's supposed to
Yep.
be one like much better than Eli Lilly or even Regeneron.
All right, this one I don't get. Which vaccines are effective with effectively zero antigen detected? I don't know what that means. Zero antigen after vaccination, but I don't know that. Why would you have zero? You need some antigen, right, to give immunity. So I don't quite get it, Sheldon. Sorry.
I don't know what they're talking about.
All right. Is it known that other contemporary coronaviruses are circulating? Do you think they maintain a consistent level of infections or come in waves like SARS-CoV-2?
No, they're circulating. You can detect them by biofire and everything. You saw them go up.
Is that a seasonal infection in temperate climates?
Yes, it's a seasonal infection in temperate climates.
Okay.
It follows the same kind of trajectory as like flu and rhino.
This is pretty funny. Can you please tell me I didn't get a Fugazi vaccine when I got J&J? I don't know Fugazi. Do you know that word? No?
Well, I guess screwed up. Is that like
which means
screwed up?
Something's damaged beyond repair. I thought it was a car dealer, Fugazi, Lincoln, whatever. Anyway,
I have no
um,
idea.
mixing
No,
and matching.
no, they're fine.
So we did a paper today on Twitter where they mixed and matched the AstraZeneca with Pfizer, and it was fine. It's actually really good, the booster
Yeah.
of mRNA. So I think this is fine. And if you got two J&J, you want to get an mRNA, that'll be fine. You just have to trick people into giving it to you, right?
I guess. I don't know. I don't know what the policy is.
Okay. Yeah. Let's see what else. Vincent and Amy and Twitter like having gold. Yeah, that and $2 will get you a subway ride. Or what is it, 275?
275.
I know everyone is on the gain of function bandwagon, but there was a paper came out a couple of weeks ago that said macrophages and DCs are not the source of inflammatory cytokines. That's not true. Of course they make inflammatory like TNF alpha and others, the IL-6. Yeah, I think they do, right, Amy?
Yeah.
You have a Right Amy shirt on tonight?
No, that's my pajama.
Oh, it's a pajama? Yeah, you can't have it. Here, that would not be good, right?
No, it's a t-shirt. It's short sleeves. I don't come to the lab in t-shirts and short sleeves.
The question I have is should breakthrough cases be studied? Their numbers are increasing and it's starting to be concerning.
Well, I don't like the term breakthrough, number one. No, they don't need to be further studied. And the numbers are going to continue to go up as more and more people get vaccinated. It's a certain probability.
Mm-hmm.
I doubt that the percentage is actually changing.
It doesn't, right? And it doesn't matter, right?
Right. But if you had 1% of 100, you have one. If you have 1% of 10,000, you have 100. So it looks like it's going up, but it's the same percentage.
And it doesn't matter if people get infected. The vaccines were not made to prevent infection; they're made to prevent disease. So don't be worried.
That's why I don't like the term breakthrough.
Yeah, exactly. And the more you check, the more you're going to see. It's natural.
Yeah, we should use the test for something smart, like how to test kids before they go to school.
Yep, absolutely. Okay, here's our friend Jeffrey. MedTwitter is confident that this increased viral load in Delta accounts for its alleged transmissibility. All right, so what Jeffrey is referring to is this paper.
Who's MedTwitter?
You know, the medical experts on Twitter.
That's what they're called now, MedTwitter? Why don't they just have their own Twitter channel then? Or a Twitter app?
You know, Jeff is hip. He knows the jargon.
Yeah, well, I'm not
so this is a paper at virological.org. Viral infection and transmission in a large, well-traced outbreak caused by Delta variant. This is out of China with assistance from the UK. So basically what they do here, they have people who are infected and they had close contacts who they quarantined, and then they check them every day by PCR to see when they turned positive and check to see when they got sick. And there are a number of things in this paper, but one of them is that they say the people infected, the viral loads and the infections were 1,000 times higher than those in earlier variants, earlier in China. Okay, that's there are other issues in this paper as well, but that's the main one. So this is PCR.
It's all quantitative PCR, right?
It does not mean
there's
infectious
no control. There's also no control that you added the exact same amount of RNA in all cases, comparing this to the previous, right?
Oh, that's a great point, because you're just taking a nasal swab and throwing it in buffer, extracting it, and putting some RNA in, right?
Right.
So you don't know how to compare them. Absolutely.
Yeah, I don't get it.
So, and it's not an infectious virus. It's meaningless.
Well, I still don't get it. It's like when they talk about this lab or this paper talks about loss of neutralization from this variant, and then 4 papers down the screen, there's another group who talks about loss of neutralization for variant B. Since it's not standardized, I don't know what it means.
Yeah. The
Okay,
bottom
then.
line here is that science is going fast and furious and loose, and you just need to be careful. But the press grabs everything like this manuscript and runs proof that it's more. No, it's not proof at all. It's just
Well, I'm
RNA.
sure that the person also had, like, something to do with it. The authors had something to do with
Do
it.
you think they had a press agent?
I think they had like a press release by their university or something, but it wouldn't be surprising.
What kind of
Look at
virus
what we just got.
causes warts?
Human papillomavirus.
Human papillomavirus is over 150 different types, and a subset of them are associated with cancers. Not the one
Yeah,
on your
but
knee.
yeah. But actually, there was a really good paper recently about papilloma and wart formation or something or other, and keratinocytes. It made me think of Saul. But yeah, it was really good. It's in my folder somewhere.
You think of Saul now and then?
Well, this was clear that this was a Saul thing.
Oh, speaking of Saul, folks, Dixon de Pommier visited us today. Amy was so happy to see him. It was amazing.
I was.
So
I
Dixon
like Dixon.
sits at a
I
desk.
miss Dixon.
He used to sit at a desk outside my office, and whenever Amy came by to talk to me, she would stop and chat with him for a bit. And they got to like each other, and now, he's been gone for a long time, and he came back. He's walking with two caney things,
Yeah, they
walking
look like
sticks.
ski poles.
And he's doing great, and he's hoping to be fully able to walk without sticks in 6 months. So he is
Yeah,
going to be joining our pods.
it's great. He's writing a book and he's super excited. And yeah, next he just needs to catch a fish and all will be good in the land of Dixon.
Where's the Wuhan Institute of Virology? You can.
It's in Wuhan.
It's in Wuhan. You can Google it. It's right there in the map. Do you want me to Google it for you? Wuhan Institute of Virology. Let's see. It should come right up. No, it doesn't come up on maps. Maybe they've censored it, right? Yeah, the map. Where's the map here? How about just Wuhan, and then we look for the Institute of Virology. There we go. We got a nice map of Wuhan, but it's in Chinese. All right,
There's
WIV.
got to be a translation button at the top of the page.
Oh, I don't need to do this. The WHO report has a map of Wuhan with all the live markets and the Institute of Virology on it. So we talked. Just search for WHO China Wuhan China COVID committee report, and you'll find it. And there's a map in there. Yeah, that's where it is. It's not downtown. It's on the other side of the river, which doesn't mean a lot.
Yeah, but that's what Koopman said.
Yeah.
Or not Koopman, Anderson.
Some cat vaxxer said it's the blurry bit on the map. Yeah, they censored it. No, but it's
Well,
in
that
the
sounds so
WHO
good.
report. This is very timely, Amy. I wonder what the real numbers of COVID-19 are, since asymptomatic people don't usually get tested. They're the biggest spreaders. So what did the paper say today that we did, Amy?
We are off by a factor of 5 minimally.
In the early days of the outbreak, right?
Yes.
Let's see. In Australia, in Sydney, COVID is hanging around here on a kind of precarious plateau.
That's because they aren't vaccinating efficiently.
Just get vaccinated, folks. That's it. That'll take care of it. I know there are countries that can't get vaccine, but the rest of you have, especially in the U.S., man, come on. There's no excuse. Based on last week's clinical updates, should the vaccinated wear masks, maintain physical distance, and other protocols to protect the unvaccinated? What do you think, Amy?
Sure. I mean,
if-
you're not really protecting the unvaccinated because they're walking around maskless anyway. So, sure.
I mean, the unvaccinated should be masked, okay? That's the primary thing to do. At Columbia, in our lab, we are all vaccinated. Is that correct, Amy? We're all vaccinated,
Yeah,
right?
every one of us
So
are
in the lab,
vaccinated.
we take our masks off. There are no unvaccinated people around. But if someone walked in and they said, I'm not vaccinated, we would
Put our mask
put
back
our
on.
masks on. I went to a meeting this week in Lower Manhattan. We all wore masks. We sat down and they said, we're all vaccinated. Is it okay with you? And boom, off come the masks. There's no unvaccinated people there. So I think you have to respect other people. So when I'm out and about, I wear my mask. And I think the public transport in New York, you still have to wear masks, I think, to protect unvaccinated people and to protect themselves, right? So everyone, they don't want to make this distinction. Vaccinated people don't have to. So they say everybody wear a mask, right?
Yeah, but what's incredible is uh, tomorrow, I think it goes into effect that, like, if you are a healthcare worker, you have to be vaccinated. At least in the city, de Blasio is having an executive order that says you
Mm-hmm.
must get vaccinated.
Some people don't want
And
to get vaccinated,
well,
maybe?
like 40% of the workforce isn't vaccinated. It's like
Crazy.
amazing.
Someone wants to know if you got tested.
Tested for what?
COVID,
Now?
because you have sniffles, yeah.
Well, first of all, I've vaccinated, so I'm not clear what the point would be. But yes, my mother made me go get tested. And the thing is,
Seriously.
yeah. But
Where'd
the
you
thing
go?
is, I went to Columbia like regular people. I work at Columbia. I went to Columbia. But the thing is that I wrote a grant. Do I not always get stressed out writing a grant?
Oh,
Hello?
crazy stress. Like you've never seen in any person before.
Right.
Amazing.
And don't I always get sick right after writing a grant when I like
Yeah,
crash?
absolutely.
Okay. So when did the grant go in?
Last Wednesday or
Tuesday.
Tuesday, yeah.
Tuesday, right? I got sick on Thursday. What do you put A plus
Well, your
B
test was
equals
negative, though,
that?
right? Your test was
Yeah,
negative. All right,
my
good.
test was negative.
Amy,
But seriously.
the wine is to protect from brain pain.
Well, send more wine then. I have a lot of brain pain. I've been, um, but yeah, okay.
UK, COVID cases are going up. They said just everybody go back to life. Okay.
Well, they have
You know,
plenty of vaccine, and it's mostly in the unvaccinated.
Yep.
If you want to risk not getting getting COVID and not getting vaccinated, be my guest. Doesn't sound like the smartest decision to me, but
Yeah.
hey.
Neva, thanks. I'm glad you like the incubator. By the way, so now, as I said, we're going to furnish it, and Amy will be the main consultant for furnishing. So it will be tastefully done.
Yeah, it's going to be good.
But as I said, those of you who come to New York, you can visit. We'll figure out a way to visit.
Well, they don't have the restrictions that we have.
You know what would be interesting is to put like 10 chairs in and have people come and sit in on recordings of podcasts.
So you want a studio audience like when they do late night with Colbert at the Ed Sullivan studio. Is that what you're saying?
Yeah, late night with Vinny and viruses, yeah.
Okay,
yeah, I think that would be fun.
Studio audience.
Can I have a shimmery curtain behind me?
Yeah, but I want you to hold the envelope up to your head while you're wearing the turban like Johnny Carson and
put
tell
it up
me
and
what's
say,
it. Yeah.
The answer is yeah,
Yeah.
absolutely. Okay.
That's what I want. And then you could have the huck or the gong for when your guests are duds. You can have the gong. I think he had a gong too. We'll have to go back and watch some Johnny Carson. He was he was pretty
Anyway,
much.
so people will be able to visit, and I hope also to attend a taping or a recording. It's not a tape, right? It's a recording.
I don't know if you have a little video to record. It's a
recording.
It's a taping.
Oh, by the
Right.
way, folks, the studio, the incubator, is 2 blocks south of Penn Station, a central hub of transportation in New York. I'm walking back from the incubator to Penn Station. There's a big sign over It's Madison Square Garden, Penn Station, right? It's all one big place.
Well, it's also the theater. There's
Oh, what is
a
it called?
theater. It's called the theater at Madison Square Garden. It's where like Billy Joel performs and stuff.
And they got a big picture of Joe Rogan, who's going to be playing live there. Joe Rogan is up there. He's such a
Okay,
star.
well, now Jeff has a goal for you to have a billboard above Penn Station saying Twiv and Vinny and friends at
But
the
he's
theater
performing
at
there.
Madison.
They'll never
Yeah,
book me.
so what?
They won't
That's
book
Jeff's
me there.
goal. That's Jeff.
That's
Now
fine.
Jeff has a goal.
I just thought it was funny because we talk about Rogan now and then on this live stream, and there he was, 2 blocks from my office. My kids' grandparents refused vaccination but have recovered from a natural infection. Can they still transmit more than vaccinated people as infections rise?
No, they're not infected anymore. It's done.
Although we do know that a boost after a natural infection gives you really good immunity. We did that paper last week, right?
Yeah, but that's not the question. Question
No, but
is, are they
they should.
shedding?
No, they're probably not shedding, but they ought to get at least 1 dose. It's too bad.
Well, they're not going to. So you have to, unless they want to read the New York Times article, which Jonathan and I decided was just too time consuming and too patient oriented to sit and have a conversation to figure out their logic of why they don't want to get vaccinated and then make an argument based on that, then they're just going to have to accept the fact that they refuse.
Right.
Not the smartest decision in the world, but everybody's entitled to make stupid decisions.
In New York City, you need 10 locks. Yes, you've seen pictures of the apartments with 10 locks, right, in a big bar. Oh, yeah.
We're not building a jail cell here, people.
No, I mean, there's a doorman, right? So you just
You'll
need
be fine.
something better than the flimsy
That's why you go to Popeye.
thing. Yeah, Popeye's going to help. Peter Open show PowerPoint thing about genetically modified. I don't know what IgM is. Do you know? Do you know what this means?
I don't know. I also don't know who Peter Openshaw is.
He's a professor of experimental medicine in England and a clinician.
What's experimental medicine? Person who runs clinical trials?
He's got a PhD and he has a lab.
Huh.
So I don't know what his thing is. JEJV. I don't get it. Jamie says, thank you for the shows. I'm more worried about people's reaction to the variants than I am about the variants themselves. Yeah, that's exactly the right position, right?
Yeah.
Just get vaccinated and you'll be fine.
And I work abroad. I wonder when I go home to visit family. You'll be fine.
Just if you're vaccinated, you'll be fine. Oh, here we go. Amy, this is for you. Is there something, materials, methods, some key bit of knowledge, etc., that doesn't exist but would it be a boon to your work if it did? Time, money, help, and wine are givens. It's great.
It's very funny.
I know what Amy wants.
What do I want?
Well, I'm not going to tell you. I think you should answer the question.
Is there something, a bit of knowledge that doesn't exist that would be a boon to my work? What does that mean? Would be a boon to my work
if you could have a result tomorrow without having to spend 6 months getting it, what result would that be?
My paralyzed mouse by EV68. My 3-week-old paralyzed wild-type mouse.
You don't want a collaborative cross mouse? Oh, that's what you're talking about.
Yeah,
right?
I mean, yeah. Collaborative cross mouse. Any mouse that I take any isolate of EV68 from 1962 to tomorrow, and I give it like we give polio, and the mouse develops paralysis 3 to 7 days later. And then I could do all of the experiments that I want. And I could look at cross-reactivity. I could look at durability. I could look at heterologous challenges. I could do pathology. You could do tons of questions. I could figure out the mechanism, the way it goes from the respiratory tract to the brain or the spinal cord. I mean, yeah, ideally, that's what I want is a mouse. Any mouse that's 3 weeks old or above that I can intranasally infect and come back and have it have, yeah.
There you go. D614G.
Yeah.
I'm waiting for someone to have alpha as a handle here, or delta, or
I
lambda.
don't know. It's too hard with all these things. Just call it one name. Then I can focus. I can't remember all these other
Amy, do you
names.
have a guess when you think things will be normal again in the U.S.?
I believe by Christmas we will all be thinking it's normal.
I think that's probably right. You
Well, I
think more?
don't think people want to have a second holiday season where they don't see their family. And that's
Mhm.
like the holiday season of holiday seasons, right?
Yes. Yes. All right, here's another for Amy. Will they ever develop a cheap lick-a-stick?
No.
My necks are costly, and I am unlikely to use them. Yeah.
No.
You
I
don't
don't
think so?
think that. No. It's done.
I think it'll be
The moment, no.
developed.
Nope. Nope. The moment has passed. The window of people's interest has passed. Now it's just wait until the kids who are under 12 get vaccinated. Nope. It's done.
Okay.
Got screwed up by a certain individual and there's no consequences. It's
So many
unfortunate.
different opinions on testing, infectiousness, vaccinations. Did you see Monica Gandhi's piece in the Post? No, I did not. Did you, Amy?
No, but I think she wrote a piece in either the Journal or the Times that I was very surprised that I thought was correct. Because normally she's a lunatic.
Is codon optimization driven only by host tRNAs, or do other considerations apply?
Well, for us, other considerations always apply.
It's the nature of the degenerate code, which codons are used in a particular organism, right?
Right.
But it can be other things, can be structures as well.
Yeah, for sure. Just think about like when Raoul moved the Cree, right?
Mm-hmm.
He had to make sure that the part of 2C that he took the Cree out of that so it wouldn't form, that stem loop or pseudoknot anymore,
Yeah.
still encoded for the same amino acids, right?
Yes. H2, thank you for your contribution. Very kind of you. Indoor plants for the incubator.
But dirt?
Well, how it's going to grow plants, right?
Oh, there's no dirt. No. We don't do dirt. We don't camp. We don't do dirt. No.
I would kill them. I would not water them.
No dirt. No
But
dirt.
if you
No
bring
camping.
one, I won't turn it down. Wuhan market mammals, palm civets, raccoon dogs mingle with horseshoe bats and mustelids. Viral possibilities are endless. No. Yeah, well, the chances of cross-contamination are quite high, as Laurie Garrett said. She described the animals stacked one on top of each other in cages. They're scared. They're excreting. Yeah, it's a mess.
Did you know I am so smart, but she's confused all the time? Did you know that?
You're confused?
Yeah, apparently Sean Diamond says Amy is so smart, but she's confused all the time. Book smart and
Well,
street smarts are different, I guess.
It's been deleted, thankfully, because it's not true.
I was like, I didn't realize I was confused. It's like
No,
I
you're not
didn't realize.
confused. I've been working with Amy for 20 years. She's not confused.
Yeah.
Until now, I thought Monkey B was a computer virus from the 90s. Now, it's a pretty dangerous
Are they
herpes
talking
virus.
about that? Are they talking? I thought that. Oh, really? They're not talking about the monkeypox case in Texas?
Well,
I
that's
sent you the
monkeypox.
article.
That's a different one, yeah.
Yeah.
This is very funny how you got the welcome to the upside down world. How did you do that? That's clever.
It's clever.
Have you read JCVI's recommendations on adolescent vaccination? I have not. You found it shocking. Are they saying not to vaccinate?
Probably. Who is JCVI again?
I don't know, Journal of Clinical Virology, JCVI.
Yeah, I've
Oh,
seen
here
where
we
they
go.
said that they shouldn't some people are coming out and saying that there's no need
Oh,
to
that's the
vaccinate.
J. Craig Venter Institute. I don't know if
Oh,
that's who he's talking about. It's not a
yeah, well,
journal.
all right.
Oh,
It's
the
8:45.
the JCVI is the Joint Committee on Vaccination and Immunization, an independent expert advisory committee that advises the UK. There you go. I can imagine what they said. I don't know what they said. No. Let's see.
Well,
JC. Go
I
ahead.
think it's the same thing that was written in an editorial by and he's an
Oh,
epidemiologist at a
yes.
Hopkins who
There we
said that
go.
there's really no need to vaccinate since they very rarely get severe disease and they don't die, blah, blah, blah.
He says they
Doesn't,
don't transmit, so they don't have to be
yeah.
vaccinated. This is the dumbest thing I have ever heard. Francois. Jeez. Oh, my
All
gosh.
right, it's 8:45. Two more questions, and it's time to go.
Here we go. Christopher, is there any credible scientific evidence published that the Delta variant is causing SARS-CoV-2 virus to be more transmissible and cause more cases of COVID-19?
No.
There is no credible evidence. Transmission is very hard to demonstrate, but what's happened, Christopher, is that the variants are more fit than the previous ones, and they outcompete them, and that gives the appearance of, to some people, better transmission, but it's not. And then people measure shedding by PCR, and as we said earlier, it's not correctly done. It's not infectious virus. It's not calibrated. So, and for as far as virulence goes, it's all the studies I've seen are not convincing that that this virus is more virulent. So, I do not see any. If I did, the day I do, I will definitely talk about it. It's not that I'm against it. It's just that I don't see the evidence, and neither does Amy. I come in every day and I say, Amy, what do you think? And she says, no, not yet.
Very much.
Do you want to have one more?
Yeah, one more.
Do you think the observed weaning, it should be waning, I guess, immunity is due to age or due to how long ago they were vaccinated?
Waning immunity is natural. So it's
Yeah,
dependent on how long ago you were vaccinated. I mean, it's probably accelerated in due to age, but
right.
it occurs no matter what. You don't want high antibody levels to remain. You want to spike and then you want to come down.
That's right.
So maybe it's exaggerated or exacerbated a little bit due to age, but no. And I don't actually think it's a problem. So I'm not really, it's not 100% clear. No, it's not Marty McRae. It's somebody else who writes for the Wall Street Journal who wrote an editorial from, and he's an epidemiologist. And I think he's some kind of MD from Hopkins or the Bloomberg School of Public Health. So also the problem with waning immunity is the fact that just as Daniel has continuously pointed out this week, and you'll probably hear a lot about it tomorrow, is antibodies are not known to be correlative to immunity, right?
Yeah,
They're not that. So unless you can demonstrate that, then I'm not really all that concerned. And as we discussed
absolutely.
earlier, right?
Hmm.
Some viruses resolution depends highly on T cells, and other viruses it depends highly on B cells, right? And
Yep,
even if you had waning immunity, which would be equivalent to being immune deficient, right? Like a B cell deficiency, those people are not being presented with more severe illness.
correct. So,
So
well,
I don't think people have really like when they talk about it, I don't think that they've processed all the information and are capable of putting all of the pieces together as we just did.
yeah, the waning immunity, if it were T cell immunity, I'd worry, but it's not. It's B cell, it's antibodies. And I just don't think that makes as much of a difference. But that's all the data people think about, Amy. And so that's what they say. Oh, it's going down. It's a big problem. But you need to think
Yeah,
a little
but
deeper.
unfortunately some of the people who've pushed this forward as waning immunity have received a lot of grant money and stuff pulling out the B cells, and that's what they and making antibodies and stuff. So it's kind of like, you know, conflict of interest.
And this is what bothers Amy.
Yes, this is part of what bothers me.
Maybe sometime you could just go till 9 o'clock. Not every time, but just one time in the future. How about that?
Well, it's like 7 minutes. I can stay till 9. It's 7 minutes.
All right.
But yeah.
Should I be scared of getting exposed to Delta if I'm fully vaccinated? It seems it might
No.
actually give me better immunity. No, you shouldn't be scared. And as Ron Fouché said, it probably will boost your immunity. But don't seek it. But you'll be fine. Although I got to say, Amy, not 100% of people will be fine, right? There will be a few in every 100 who get very sick, even though they're vaccinated. But that's because the vaccines are not 100% protective, right?
Yes. Well, nothing in this world is 100%. I mean, the one
Amy
process
is
that
100%.
the one process that needs to be 100%, which is reproduction, to continue the race, right? To
Yeah.
continue the species, is not 100%. So why would I think that anything else should be 100%? I mean, come on.
The only thing that's 100% is death, Amy.
Well, yes, but that's not to continue the species.
Yeah, okay. I know you talked about a universal flu vaccine on 12 months ago. When will that actually be able to be at the local pharmacies?
15 to 20 years from now.
Really? I think 10. But she's right. She'll be right. You'll see.
I just think that there's going to be a lot of fail.
Oops. Amy just froze.
That they're going to reassess and try and make an mRNA vaccine.
I agree with you.
So you'll have to start all over again.
I agree. I did a gain of function in my bedroom, transforming bacteria with GFP. Yeah, that's right.
How did you do that in your bedroom?
You go to one of these places where they teach you how to do science at home. That's what they teach you how to do.
Oh, really?
A few weeks ago, Vincent said making cheese is gain of function. Mind blown. If so, gain of function is delicious and my favorite food. Well, what I said was if you modified the yeast, say, to make a different tasting cheese, that would be gain of function, yeah. But, when
I think they modify molds,
and they mix different ones together and do, yeah, they probably are changing their properties as well. Could you try again on gain of function? Sounds like what they were doing was loss of function. Not in the experiment that Rand Paul was talking about. It was no change in function. You're just substituting one ACE2 binding spike for another. I'm guessing that lots of advances in genetics have occurred since Paul was in med school. Yeah, but he may not have learned any of them anyway because they don't teach you much genetics in med school, right, Amy?
Well, I think he was just absent on those days. And it doesn't matter if there's been a lot of advances in genetics. There's fundamental principles that have remained the same since the P-man, Mendel.
Yeah.
And stuff. So gain of function was defined at the time of the P-man. So I don't know why that would change.
No, that's true. Gain of function. The only change, Amy, is the use of recombinant DNA to modify organisms, right? And
yeah, but that's a detail.
Yeah, but Paul doesn't know about that.
But the principle of gain of function mutations were observed with the pea plants and the fruit fly eyes and Punnett squares and blah blah blah, and all of that. Even though it turns out that he kind of made up the numbers. Mendel kind of made up the numbers. Turns out that while he kind of made them up, he was right, and so therefore, those are fundamental principles. I just don't get it.
I got COVID. Wife and I both vaxxed. Is there a point to quarantine at this point?
Well,
Nope. Do your thing. Oh, yeah, this is a good one. Lewinsky's, that's actually Lenski. Robert Lenski's long-term evolution of E. coli is an interesting gain of function. It gained the ability to metabolize something. Yeah, he just passes it in the lab. It's done like 60, 70,000 generations.
yeah.
And yeah, it changes. It
Yeah,
changes.
but this is the classical definition that Fauci was using
Yes.
by
I
serial
understand.
passaging.
Why are the Academy of Pediatrics and Tony Fauci saying even vaccinated children need to wear masks next term? What is your understanding of that, Amy?
There will be a significant number of children who will be still beneath the age of vaccine.
Yeah, so they're going to school, and so in the close contact, right? You can't keep them 6 feet apart or whatever, so that's the recommendation. Yeah. Given that SARS will be endemic, will most unvaxxed people get infected?
Well, you'll
so.
get infected as a young kid. It'll be a childhood disease.
But the adults who are currently unvaccinated, eventually they will encounter someone with virus and they'll get infected. Yeah, I don't know how long it will take to get everyone infected, but yes. And then as kids are born, they'll get infected early on. And that'll be the cycle, I think, right, Amy?
Yeah, I think that'll be the cycle. And I think, like, we see it now, right? That the adults who didn't get vaccinated for whatever reason are paying the consequences. Listen to Daniel. It's very upsetting.
Which vaccine is best at promoting IgA? It seems like that would be the best at preventing infection with a respiratory virus.
I believe that Michael Diamond's intranasal vaccine is the one that promotes the best IgA development. And then the role of IgA was described by Theodora in a paper. There was a back-to-back paper about dimers of IgA.
The problem with Secretary IgA, and Amy's right, a mucosal vaccine, so flu mist, which you spray in your nose, is also good for secretory IgA. The IgA that you get from an intramuscular vaccine is serum IgA that goes up through the epithelium and goes into the mucus. It wanes quickly, and so unless you have a lot of memory IgA producing cells, you're not going to have a quick memory response. And so many respiratory infections occur, and because we can't mount a good IgA memory response. And whether Michael Diamond's vaccine will do that is a good question.
Yeah, I don't know. But generally, respiratory viruses are poor. You get a lot of them. And generally it has to, and don't always get memory or good memory. And a lot of times it has to do with the fact that there's no viremic phase to them. Remember your table? Where's your little book thing with the table?
I have it. You want me to show you? It's really cool. I did it today, actually.
Yes, and did you tell them? Did you tell your class who was the biggest proponent for this? And then somebody from the NIH took it over?
I did not, no. Which lecture was it? Can you remind me?
I don't know.
It was adaptive immunity. Here we go. I made a nice table. Here it goes. Here we go. Let's show this before you leave. So this is...
Gotta make it bigger. It's still too small.
Okay. Well, I don't think
now it's good.
That was good?
Yeah, it's good.
You can see that? Really?
Yeah.
All right. So
no,
here we have
I make it up.
disease. So these are systemic infections, right? Viruses that spread through the blood to other places. Look at how long antibody persists. This is after infection. This is not vaccination. Look at the 75 years for vaccinia. Wow.
65 for measles, 40 for polio, 75 for yellow fever.
All right, then you look at mucosal infections. Pff, look at how anemic they are.
Yeah,
So
they're wimpy.
they're very wimpy. So we have to overcome that with a vaccine in some way. I don't know how we're going to do it, but
Well, you have
and
to first acknowledge the problem.
indeed, indeed. So Amy had this
Because
idea years ago, many months ago, that viruses that cause viremia will give you a really good memory response. So what do we do? Make a vaccine that goes systemic? No, we just have to make the immunization, the IM, or maybe the nasal spray somehow make better memory. We don't know how to do that.
right now it's 9 o'clock. Now I have to go.
One more here. If you
All right,
stopped
one more.
IPV, would the disease be back?
If you stop IPV, will polio be back? Yes, for sure. And you see that in Africa where they have been unable to complete rounds of IPV, or Afghanistan and stuff, and then they have big outbreaks of polio, yeah, and stuff. But I mean, I know you and Alan think that we're going to stop vaccinating, but I don't think we're ever going to be able to stop vaccinating.
I hope I live to see the day.
You think it's going to be in the next 30 to 50 years? I don't think so.
I just said I hope I live to see the day. I don't know, right?
Yeah, I think that's way too early. I would not expect it to be for more than a century from now.
Wow. Oh, I got to say,
Because
Amy,
how
you're
do you
often
know
right.
how many people do you know? We're not really monitoring, right? And we're not or shedding, right?
Right.
So maybe there's somebody who was immune compromised who was one of the last people to get OPV in 2000. They're going to shed for, what did Adams' papers go? Just was one person for 28 years, right? That's all
Yeah.
he's.
Yeah, sure.
So, right? You don't even know it, if you continue to follow that person, if they'd be shedding even today, right?
Yeah, of
Which
course.
is probably another 7 to 10 years after Adam did his study and finished tracking the person, right?
Right.
So right there, you're at 20 years. And
You're probably right.
if you were a kid, you're only 20 years old. You're going to live another 80.
Mm-hmm.
And then,
Okay.
yeah, I don't know, whatever.
All right,
All
Amy,
right.
thank you again.
I'll see you tomorrow. We have to do the IRB. It got sent back to us. We have 3 things to fix. And then we could collect
Okay.
samples. I'd be super excited about that.
Okay. Thank you
All
again.
right.
Good night.
See you tomorrow. Bye.
Bye-bye. All right. It's been suggested, excuse me, that the variants evolved in single individuals with a long-term infection. Right. That's because early on, last year, some individuals of this sort were studied with long-term infections, and they had highly mutated genomes with some of the changes that are now seen in the variants. And I'm not sure that that is the only place they evolve now. I think they arise in other individuals. I think some of them may arise by recombination of variants with fewer changes and so forth. You say, would you expect to find changes in T-cell epitopes? Yeah, so that's a good point. So I don't know if the 130 days or so would be enough to give evolution of T-cell epitopes. But, they may not have an advantage in the next person. They may be neutral and just go away also, right? The T-cell epitopes are only good, the changes in T-cell epitopes are only good for you to match your MHC. So I'm not sure that would be the case. Thank you, Les. I appreciate your support. Yeah, they are infectious, aren't they? Is there a link between blood type and COVID infection? Funny you should ask. I just gave a lecture. I'm teaching a summer virology course at Columbia. Today was the pathogenesis lecture, and I showed a slide which shows that there's no link between blood type and COVID infections. Are there links for other viruses? Yes, there are. I think one of them is a norovirus link, but I'm a little hazy on the details there. I'm just a soul whose intentions is good. Please don't let me be misunderstood. Yeah, that's a song. I don't know who did it, though. Tell me who did it. I know. Is it the Doors? No. All right, here we go. Seems to me that any breakthroughs for long hauler treatment? No. It's going to be a while. It's going to be years. It will take some time. Ralph says, it seems to me there are multiple meanings of gain of function. Mark seems to specify dangerous GOF. Well, what does Mark know what's dangerous or not? He has no clue because he doesn't know what the benefit of gain of function experiments are. So I don't take his classification of gain of function at all. But the problem with him saying that is that the press thinks that all gain of function is dangerous, okay? Because they don't listen to the details, and that's what the press thinks. Anything GOF is a bad word. It's not. I would have done your studio differently. Take out the tiles and lights, install a lighting grid. They wouldn't let us take out the tiles. I wanted to have a bare ceiling where I could put, pipes in to hang lights from. I agree. But they didn't want to do that. The building owner, the guy who owns the building, he wants to keep it, his way. But I tell you what, Dennis, you're right. I like the idea. If the whole venture is successful, I would consider paying to take out the grids and put hanging lights in it, like a proper studio, right? Yes, I agree. I find it weird that the press points out how older people are getting COVID while being vaccinated. Well, COVID or infected, that's the key here, right? The vaccines were very good at preventing COVID, the disease, in older people. And, part of the confusion is that people think COVID is the virus, right? So if they get sick, and as I said before, no vaccine is 100%, so someone's going to get sick. The older are more likely to get serious disease. So it all fits in with that, I think, right? I don't think that's an issue. So enhancing the binding capability that's already present is not gain of function because there's no new property. Right. There was no enhancement of binding. The previous bat virus could bind ACE2 and get into human cells, and the recombinant they made could also bind ACE2 and get into human cells. So no net change in property, no gain of function. It's a very subtle thing, right? Especially for a senator. Not to impugn any senator. Listen to the scientist. Who's the scientist? Tony Fauci. Or you could listen to Twiv. I've looked at gain of function as a genetic mutation or just mutation. You don't have to say genetic mutation. That enhances the function of a gene. Changes it in some way to give it a new property. That's the key. It's a new property. You could change a gene and not have a change in a property, then it would not be gain of function. Or you could change it and it would. That's what I just said. Sorry. Art for the incubator walls. Just Google your photo on canvas print. Yeah, that could be fun. I got actually a. Let me show you this. I got a watercolor done by an artist who I very much like. She is based in Washington. Her name is Artologica. You can find her on Etsy. So I commissioned a really big watercolor, which I'm going to put behind me in the incubator. And so isn't that cool? I had her use the Big Sur palette, the Mac OS. Because I really liked the bright colors of that palette. And she loved it, and I said, just make whatever viruses you want, including fanciful ones. My daughter said, what is that, a sperm? I said, no, it's supposed to be a virus, but it just doesn't exist. Okay, but I like that idea. That's good. Okay, so is the R0 of Delta higher? So the R0 is a mixture of viral properties and people's behavior, right? Now, they recalculate the R naught often, and the UK PHE often does this. I really have an issue with them. They recalculate the R naught without taking into account human movements and interactions. So it's wrong. It's not a properly calculated R naught. And you know what? R naught is not really a constant. It changes depending on interventions, where you are, and so forth. A better indication of people's movements and interactions is a secondary infection rate. And guess what? Public Health England is saying it's going down for Delta. So if Delta is biologically more transmissible, the secondary infection rate shouldn't decline. It just tells me that the increase they saw was all due to human interactions, not to the fundamental properties of the virus. What the virus is, what the variant is more fit, so it displaces others. So that's why it's 80% in the U.S., right? It's not surprising. It's what flu does every year. A new variant arises that displaces others. We never call them more transmissible. We never say they have a higher R naught. As Ron Fouchier said, figuring out the R naught is really hard because you can never account for the human movements. And now that the population is partially immune, you're supposed to calculate R naught at the beginning when there's no immunity. So now you do something that's not really R naught. You do an RT or an RE, but you still have to calculate the effect of people, and they just do not do that. And so if they're mass gatherings, yeah, you're going to see a higher secondary infection rate because there are more people together. They never account for that. They only look at the rate of new infections. And I think that's not correct. And many people agree with me, including Ron Fouchier and others. So it's not like I'm a whack job. I'm not a Peter Duesberg. Was that his name? Yeah. Who doesn't believe that HIV causes AIDS. I'm not a COVID denier. I just want to see the data for the R naught. Okay. Engineer a virus to mine bitcoins. Boy, that's a computer thing, right? Just use a lot of electricity. I work in a hospital and my COVID patients have high ferritin. What's the connection? I do not know. I have no idea what the connection is. I will ask Daniel. Oh, here we go. Patricia says ferritin's an acute phase reactant. So you have inflammation, an infection, and your ferritin goes up. Okay, thank you. Yeah, Delta is RO8, according to Paul Offit. He's wrong. Now, I like Paul a lot, but he's wrong. It's not 8 because all he's doing is repeating what Public Health England said, and their calculations are wrong. It is not 8. It is 2 to 3 until proven otherwise. And it just goes to show I'm not a, what did someone accuse me of being? Surrounding myself with people who say what I like to hear. So Paul Offit is wrong about the R naught, but he's just repeating what he hears. If we can make enough immunity in the population to make COVID outbreaks small with mild disease, that's a massive win. Yes, and that can be done, right? It's just we need to vaccinate more people, but it becomes a political issue, and you can't get everyone vaccinated. And there's no way to force them. I don't think you should force them. We're stuck with it. That's humanity, right? Some countries, everybody gets vaccinated, but others, they don't. Is there a way to check one's level of immunity the same way we check with an oximeter? Not at the moment, because you have to draw out serum and look for antibodies in that. So, I mean, it's not a big deal. You just take a pin and prick your finger and get a drop of blood. That's enough to do it. But it is a little bit more involved, right? It might be possible one day, though. Lynn, thank you so much for your support of our work and the Incubator. Why do some say this will mutate to something akin to the common cold? It's just wild-ass guessing. Well, it's not going to be deadlier. Why would it be deadlier? There's no, as I told my students today, there's no selection for being more deadly. It doesn't benefit a virus to kill its host quicker. It could be a side effect of some other necessary change that's selected for, right? But not to kill it faster. So I don't think it's going to get more virulent, and I think the reports of increasing virulence for that reason are nonsense. Anyway, the data are not good. Why do people say it'll be like the common cold? The idea is once the population is entirely immune, then only young kids will become infected, and they get less disease than adults. We already know that. Not zero, but less. And that's what happens with common cold coronaviruses. That's the idea. It is a guess. Absolutely. Absolutely. It's a guess. Thank you, Lynn. I appreciate that we're of use to you. Fugazi is a fake diamond. Okay. Thank you. My understanding. Oh, here we go with the clarification. As many vaccines eventually no longer let you detect antigens. Yes, of course. Just like a virus infection goes away eventually, vaccine, the antigens are gone, right. And so what you have left are memory B cells and memory T cells that remember their encounter with the antigen and can respond again when you're infected. So I don't remember your question, but maybe that helps. Why does our body attack the spike that mRNA vaccine induces to make? So all proteins that we make are attacked, as you say. They're not really attacked. They are sampled. We have cells that patrol us, dendritic cells, macrophages. They're called sentinels. They are always picking up proteins and that are floating around in your fluids, bits of dead cells, and sampling them to see if they're foreign. And what they do is they bring them into the cell, they digest them, they put them back on the surface, and they go into a lymph node and present it to a lymphocyte, a T cell. And if the T cell matches the peptide, it means it's foreign. And then they will make an immune response. And so that's what's happening. They're not really attacking it because they're just sampling it, and they're sampling all proteins that are made at any given time. What's known about HKU1? I don't remember what specifically HKU1. It's been around for quite a while. It's I don't remember if it's one of these that arose 100 years ago or hundreds of years ago. I just don't remember but most of these common cold coronas are thought to have emerged from either bat or rodent or cow reservoirs, some other animal, anywhere from 100 to hundreds, 800 years ago for some of them. So they're all zoonotics originally. And they may have caused a big outbreak initially. We don't know because we don't have good records of outbreaks, before the early 1900s. And so if it did cause, say, a pandemic, now it's causing a common cold. That's part of the reason these 4 common cold coronaviruses cause mild disease. We think they spilled over and may have been serious at that time, and now they're moderate. That's part of the reasoning for this wild-ass guess that SARS-CoV-2 is going to become another common cold coronavirus. Have I heard of Descript? I have heard of it I've seen some YouTube videos where they say it's the best, easiest way to edit videos. And I've found in my experience that easy is not always the best, right? I think some things you have to put some sweat into and then you get a good product. So for audio editing, I use Logic Pro, and for video editing, I use Final Cut Pro. They're both Apple products. I like them. They're not perfect, but I've learned how to use them so I can move relatively quickly. I would like to try some others. There are equivalents from Adobe, which I actually own. I've never tried because I don't have time. There's DaVinci Pro from Blackmagic Design, which is really cool, but I don't have time to learn them. I'm hoping once the studio gets going and I have people helping me, I can put some time into other things. I'm glad you look forward to it. I appreciate that very much. I do too. I very much look forward to doing this with Amy. So does Amy, by the way. The RSV uptick in the Netherlands, so respiratory syncytial virus, is explained by less infections in women during lockdown. Oh, now that lockdown is relaxed, there are less antibodies from mother to child at the moment. Yeah, that makes sense. Yeah. Did you read that somewhere? Is there a paper? I'd love to see it. I'd love to see it. What's the CureVax change? They use non-modified bases instead of modified bases as in the Pfizer and Moderna products. Could you explain what VAERS vaccine? What does it stand for? VAERS. Vaccine Adverse Event Reporting System. It's a system set up in the U.S. It's co-managed by CDC, and you can report an adverse event of a vaccine to it. So, this is used by anti-vaxxers to say that vaccines are not safe, right? The problem is, if someone gets a vaccine and the next hour dies, they may have been hit by a car or something, right, or died of some other natural cause. So it doesn't mean that these events are caused by the vaccine. It's just they're associated. That's why it's an association until they investigate them, and most of them end up not being vaccine associated or caused by the vaccine. So that's what it is. You did the AZBN combinations on TWIV today. Yes, I'm going to edit it after this and release it at midnight. That's right. Do vaccines prevent long COVID? Still too early to tell. I asked Daniel that last week, and he said, still too early to tell. You know what's cool, folks? We have 566 people watching. It's up from last week. It's so cool that people want to listen to virology. In your lectures, you mentioned that healers don't have ACE2. Are there people that also don't have ACE2 and are therefore naturally immune? I don't believe so. I believe ACE2 is essential. And there are people that don't have CCR5, the HIV co-receptor. 4 to 16% of people, mostly of European origin, have a deletion in that, and they don't get HIV as a consequence. But I'm not aware of any for ACE2. All right, here is my buddy Renzo Dalla Via. Come stai questa sera? I assume as time passes and as you all have discussed with lowering of antibodies and reliance on T cells, there will be more people showing. Showing what? Symptoms? Mild symptoms? Media doesn't get it. Yeah, so I think we'll get to a point where the disease in fully vaccinated people gets more serious from year to year. That's what happens with flu. There's disease, as the antigenic variants emerge and they evade your immunity, the disease gets slightly more and more serious. Now, that's flu. It's not SARS-CoV-2. But it may be that when the variants can evade antibody up to a certain amount, we don't know what that point is, then we get more severe disease, and then they may have to change the vaccine. And it won't be a booster. It's going to be a new vaccine. I don't know when that's going to happen. Some people think it's sooner rather than later, but I don't think so. If we can cut down the number of global infections, we could cut down the generation of variants. So it's a race, right? But I think for the moment, we don't need new vaccines, but it may be we need one in a couple of years for that, because we don't know what protects you. Is it antibodies, T cells, or both, right? We see antibody levels waning, but we don't know what that means. Because even with antibodies waning, people still don't get severe COVID after being infected with a variant and after being vaccinated, because probably the T cells are protecting you. So do you need a little bit of antibody protection? We just don't know. That's what a correlative protection is. We need to know that. If this study performed the study statistically, then it's true. They didn't, Henry. That's what I'm saying. They didn't quantify the amount in the samples. End of story. Can Binax now pick up an asymptomatic infection in a vaccinated person or a person that has recovered from a natural infection? Yes, it should be able to. It should be sensitive enough, even if there are low levels of virus reproduction in them. Yep. Can the Delta variant cause more permanent organ damage than the initial variant? Not that I'm aware of. No reports of that, no. There is not a global consensus in the scientific community. It's amplification of bad data. So you're saying to me, John, that I'm wrong and all my colleagues are wrong. Well, we could be, but there are no consensus doesn't drive science, John. Data does. Yes, well, this is part of the problem. It's not just the media, but it's health departments, right? They just amplify it because here's how it works. Public Health England gives a report saying the R0 of Delta is 8. Everybody just repeats it without looking at the data. Did you ever go to the data, any of you, and look at it? You're not going to be convinced. Nope. What happened to Dixon? He had back surgery, so he's just recovering. Why do mRNA vaccines cause myocarditis? Unknown. May have to do with the spike protein causing syncytia in the heart cells, but COVID also causes myocarditis at a higher level, and so that may be giving us some clues. Real life story. My wife's colleague didn't get the vaccine because he was told COVID vaccine will make him infertile. Now he's in the hospital and begging for a vaccine. Yes, Daniel gives us these stories also. Someone said she didn't want the vaccine because it had a microchip in it. It's sad because the right information is out there, right? I have antibodies from COVID infection. How is natural immunity holding up? It's not really been studied in the way that you'd like. So the vaccinated population has been best studied for that. But I can tell you this, if you get an mRNA one shot, you'll get an incredible boost. It'll broaden your activity of antibodies against all the variants. So it's really worth getting. All right, so important, John, cases increasing. No, what you're saying is infections are increasing. Time to get rid of John, folks. Not doing any good here. Are there any updates on mRNA vaccine trials for non-COVID viruses? No, but there's a malaria mRNA vaccine that was tested in mice. We did that on TWIP some time ago. So, Pam, your son is afraid of the vaccine making him infertile. That's just totally bogus. That doesn't happen. You can tell him that a virologist of 40 years experience, plus a clinician of many years, Daniel Griffin, says that. A cardiac inflammation, Daniel Griffin says there's a greater risk from cardiac inflammation from COVID, so it's worth it. And you can treat it. That's the key. You can treat it. Does anybody still work on rabies? Absolutely. Is there anything left to learn? Well, we don't really know how it makes the disease that it causes, right? So people still try and understand how the virus gets in from a bite into the central nervous system and what it does there. Quite a few labs here in the U.S. and in Europe are working on rabies, yeah. Is antigen expression inside host cells regarded for good T-cell response? So T-cells will recognize antigens that are presented on the surface of dendritic cells and macrophages, right? They don't take up antigens themselves. So they either have to be, they can be soluble proteins taken up by antigen-presenting cells, or so you get the Novavax, it's a protein vaccine. The sentinel cell can pick it up, can phagocytose it and present it. Or if it's an mRNA vaccine, the mRNA can get into the dendritic cell or the macrophage and translate it and process the protein. All right, so let's look at in these last few minutes. By the way, one night I will keep going until everybody leaves. I'll do that at some point in the future, but not tonight because I have to edit TWIV. Thank you, Zachary, for your contribution. Proof of vaccination, no mask, safe to go. I'd go, for sure. Absolutely. I appreciate that contribution. Let's see. And, as always, we have so many questions. By the way, next week, Daniel is going to do a special episode to get through a lot of the backlog of questions that you've sent in. That should be fun. Super chat from Tom. Thank you so much. Really appreciate it. Ditching the Grind, thank you so much. Really appreciate these contributions. All go to running the incubator. which is a big experiment, right, in science communication. A lowly professor tries to start a communication company. We'll see. John, thank you so much for your contribution. Sandy, thank you. Really appreciate it. Buzz Earwave, that's a new one. Thank you for your contribution. And Sean, I'm familiar with Sean's name there. Whoa, don't let me be misunderstood by the animals. Right. I have all these old songs in my head. You know, last week on Twiv, I was talking about Mountain, right? And Anthony from Jersey City, who sometimes is on this live stream, he said, boy, I didn't think anyone listened to Mountain. Or Scott Muni. Whoa, Scott Muni. I used to listen to him as a kid. All these names from the past. Thanks, Morris. Really appreciate your contribution. Also, Ian. I sent Ian the op-ed. If you want a copy of the op-ed, Vincent at microbe.tv. And I'm getting ready to send out the pre-op-ed that the Times didn't publish, whoever wants that as well. I'll send that out to you. Thanks, Ralph. Just want to say to the anti-vaxxers in chat, find some other playground. I like being around people who believe in science. We do. Data-driven. Thank you, Aaron. How do memory B and T cells remember? Well, they stick around. There are just a few cells that stick around in the bone marrow or in the circulation or in tissues, and they are ready to go when antigen comes back. So the cells never die. That's the end game there. But exactly how they do that, well, they don't not die. They divide very slowly. But certainly more to figure out there, that's for sure. Thank you, T. Klupper. Appreciate that everybody comes here. Thank you for your contribution, Overjoyed. Anything, a buck is fine. Anything you can do is great. We appreciate it. Believe me, and I know I don't thank everyone individually who has helped us on Patreon or PayPal. I hope to get better at doing that. Amos, thank you so much. Really, really appreciated. Yeah. That does it for tonight. I have to sign off now and edit tonight's TWIV so you can have it by midnight. And, yeah, Frank knows all about Mississippi Queen. I like Nantucket Sleigh Ride. It kind of ties into last week's TWIV. I could keep talking to you folks forever, but time to go. Right, one more. I had a splenectomy. Got Moderna. Should I be concerned? You have other lymphoid organs. Should be okay, but you might want to get an antibody test or a T-cell test or both. Thanks, everybody, once again for joining, and we'll see you next week. Please bring your questions back next week. Bye-bye.